Hi everyone, welcome to Pharm.D Guru. In this video, we will discuss about inhibition and induction of drug metabolism. First, let's talk about induction. Enzyme induction is a clever way your liver increases the speed of drug metabolism. So, in normal conditions, for a drug to be metabolized by liver, let's say it takes 2 hours. When this concept of enzyme induction happens, the drug will be metabolized within 45 minutes. Let's explore what happens step by step. Okay? This whole concept of drug metabolism occurs in livers, right? So, all these enzyme induction and inhibition all occurs in the liver. Remember that. Okay? Let's take a single liver cell, which is also called hepatocyte. Inside this liver cell, we have enzymes. These enzymes have active sites where a drug or chemicals binds perfectly into the enzyme's active site like a key into the lock. Here, drug molecule or chemical is called a substrate. These enzymes then does its job, which is changing the substrate into product. Once done, the product leaves and the enzyme is ready for the next drug molecule. Got it? Let's see an example for better understanding. Suppose you drink a glass of milk. We know milk has lactose. So, inside the body, we have an enzyme called lactase. So, this lactose is a substrate here. And this lactose binds with active site of enzyme, which is lactase. Now we have this complex of lactose and lactase. Then lactase, which is an enzyme, does its work, which is breakdown of lactose to products like glucose and galactose. Later, they leave the active site and the new molecule of lactose binds with the enzyme lactase. This is how normal enzyme process happens inside our body. Now, let's see what happens to this process during enzyme induction. Some drugs often does not bind directly to the enzyme, but instead they bind to the receptors inside the cell, sometimes in the nucleus also. This receptor acts like a messenger and sends the message to the cell to make more enzymes. The message reaches to the nucleus and later production of mRNA increases, which ultimately increases production of enzymes like cytochrome P450. So, more enzymes means drugs are metabolized much faster than the usual. Important point here is if patient take drugs along with this drug which has enzyme induction property, we know it increases the enzyme number inside the cell. So, those new drugs also gets metabolized much faster. Example, phenobarbitone is the drug which has this enzyme induction property. It binds to a receptor inside the liver cell. That receptor signals the cell to increase production of cytochrome P450 enzymes, leading to more enzyme receptors are ready to handle drugs. If the patient take another drug like warfarin or phenytoin along with this phenobarbitone, they also gets metabolized much faster. This means warfarin or phenytoin leave the body much quickly. So instead of 6 hours, they show their pharmacological action like only 4 hours. So the doctor has to raise the dose or increase the frequency. That's why when phenobarbital is started for a patient, doctors monitor warfarin and phenytoin closely. They know the new enzyme workers, which are cytochrome P450s, are working over time and could clear these medicines too fast. Okay, let's see some real life example. Phenobarbitone, a drug used to treat seizures, tells the liver to make more enzyme work. Result: not only does phenobarbitone gets cleared faster, but so do other medicines like phenytoin, another seizure drug, and warfarin, a blood thinner, or even itself, phenobarbitone. Carbamazepine and rifampin do the same thing. Rifampin is so strong it can make the body clear another drug, midazolam, up to 20 times faster. Cigarette smoke can double how quickly your body processes theophylline, which is a drug used for breathing problems. Smokers might need much higher doses to stay effective. Let's see another mechanism involved in enzyme induction, which is hormone-induced cytochrome P450 expression. This means certain hormones, when enter the liver cell, they bind to receptors inside the liver cell and signals the nucleus to produce more mRNA. More mRNA means more enzyme production. More enzyme production means the speed of the metabolism process. Got it? Now let's see this mechanism in molecular level. Okay, some hormones and compounds from cooked food like aromatic amines present in grilled meat or cigarette smoke chemical interact with the receptors. Here the receptors we are talking are AH receptors. AH means aryl hydrocarbon receptor. This hormone receptor complex travels into the nucleus of the liver cell. Inside the nucleus, the hormone receptor complex pairs with another protein and binds to a specific DNA region called the xenobiotic responsive element or XRE. This triggers mRNA production, which later translates into cytochrome P450 enzyme. These enzymes are then sent to the membranes of the endoplasmic reticulum, where they metabolize drugs and other chemicals. These hormones increases enzymes like cytochrome P450 levels. These enzymes acts on drugs like tamoxifen, dacrin, acetaminophen, and xenobiotics like dietary phytochemicals and carcinogens like aromatic amines produced in cooking and chemicals found in cigarette smoke and produces enzyme inductive effect. Got it? Now, it helps us understand why some drugs like tamoxifen or acetaminophen might be broken down faster or slower depending on hormone levels or exposure to things like cigarette smoke. Now let's look at the third mechanism of enzyme induction, which is nothing but induction by inhibition interaction. If you look at the structure of liver enzymes, particularly this cytochrome P450 family, we can notice it has a heme group. What it does is it plays a crucial role in detoxification process. Okay, this heme group is an essential prosthetic group which is vital for drug and chemical detoxification, as well as some other chemical reactions like catalase, peroxidases, and nitric oxide synthase. Okay. Now, certain drugs or chemicals like isoniazid and macrolide antibiotics, ethanol, and even some xenobiotics. Here, xenobiotics are chemical substances, often man made. They are foreign to the organism's biological system and are not produced by the body. Example: substances added to the food, such as flavoring and preservatives, and compounds in cosmetics are considered as xenobiotics. Okay, so these drugs, isoniazid or macrolide antibiotics or xenobiotics, etc., they all bind to the heme part of the enzyme and causes temporary inhibition of enzyme. So the liver senses this inhibition and increases the enzyme production to compensate the loss. Now we have more enzymes, and most important is the newly formed enzymes are more stable ones, so they last longer and metabolizes these drugs and the drugs taken along with it. The mechanism involved here is liver senses the inhibition, so it orders nucleus to make stabilized mRNA. Now, with this stabilized mRNA, more enzyme proteins are formed. These newly made enzyme proteins become more stable and they last longer, which is called post-translational stabilization. Now let's see some real world examples. Okay, isoniazid induces cytochrome P2E1 this way. Macrolide antibiotics induces cytochrome P3A1 this way. Various xenobiotics also use these complex mechanisms to induce cytochrome P2E1. Now let's see one interesting mechanism here. Until now, we only talked about heme group. Now let's talk about a concept without a heme group, which is apoprotein. They are more accurately referred to as apoenzymes, which is the inactive protein component of an enzyme that requires a non-protein cofactor such as a heme group for its function. If you consume low concentration of ethanol, the ethanol stabilizes the apoprotein form of cytochrome P2E1, the enzyme without its heme group, and the ethanol protects this apoprotein form of cytochrome P2E1 from degradation. So the enzyme stays longer. What it means? It metabolizes more drugs or chemical molecules. In this type of induction, more enzymes are available to metabolize drugs, chemicals, or toxins. But at the end, they generate products which are reactive in nature, which increases a risk of liver stress or toxicity, especially with substance like ethanol. Now let's talk about the second major part, enzyme inhibition. In enzyme induction, enzyme activity or count increases, leading to faster metabolism. Whereas in enzyme inhibition, the opposite happens. Enzyme activity decreases. This slows drug metabolism and can cause drug buildup in the body, resulting in serious or even toxic effects. By definition, enzyme inhibition is the decreased ability of enzymes to metabolize drugs due to interference from another drugs or chemicals. When two drugs are taken together, one may bind to enzyme's active site, blocking the other drug from being metabolized. If the second drug has a narrow therapeutic index, this buildup can cause severe toxicity. There are two types of enzyme inhibition: direct inhibition, indirect inhibition. In direct inhibition, the inhibitor directly interacts with the enzyme and blocks its activity. Whereas in indirect inhibition, the drug decreases the total number of enzymes, either by inhibiting enzyme synthesis or by promoting enzyme degradation. Now let's understand direct inhibition in detail. There are three main forms: competitive inhibition, non-competitive inhibition, and the final one is product or feedback inhibition. Let's see competitive inhibition. In this type, two drugs compete for the same active site on the enzyme. Only one can bind at a time. If the inhibitor binds first, the other drug cannot. Example, methacholine and acetylcholine. Both resemble each other and compete for the enzyme cholinesterase. Here, methacholine acts as an inhibitor and prevents acetylcholine from being broken down. Since both compete for the same site, it is called competitive inhibition. Let's see the second one, non-competitive inhibition. Here the name itself says there is no competition, but instead the inhibitor drug binds to the enzyme at a different site, not the active site. This binding causes the change in the shape of the enzyme, making it less effective. So, at the active site, even the second drug bound, the enzyme does not work on substrate. Example, isoniazid inhibits the metabolism of phenytoin via non-competitive effects. So, If the patient takes isoniazid and phenotide, even the phenotide drug molecule binds with enzyme active site, isoniazid bind to the enzyme at a different site and make the total enzyme inactive. Since no competition for active site, this type of inhibition is called non-competitive inhibition, and some other examples are metals like lead, mercury, arsenic, or organophosphorus insecticides can non-competitively inhibit enzymes. Now let's look at feedback inhibition, which is also called product inhibition. In this type of inhibition, a drug molecule comes inside the liver for metabolism, binds with an enzyme active site, and the enzyme processes this drug and finally releases the end products. But here is the problem. The shape of the substrate, which is the drug molecule, and the formed product looks similar. Since both have the same shape, instead of substrate, this product binds with the enzyme's active site. This type of inhibition caused by product, no? So it is called product inhibition or feedback inhibition. Example, allopurinol inhibits xanthine oxidase. Generally, hypoxanthine converts to xanthine, and later this xanthine converts to uric acid. High amount of uric acid in our body causes gout. This is the process that generally happens inside our body. Here, hypoxanthine converts to xanthine by the enzyme xanthine oxidase. We know allopurinol is the drug to treat gout, right? Allopurinol is designed to look very similar to hypoxanthine. When allopurinol is taken, it tricks xanthine oxidase. Instead of hypoxanthine, allopurinol enters and binds the active site. Xanthine oxidase tries to process allopurinol, just like it would with hypoxanthine. The enzyme converts allopurinol to oxypurinol, which is the product here. This oxypurinol, which is the product made from allopurinol, binds very tightly to enzyme's active site. As long as allopurinol is stuck in the active site, new xanthine or hypoxanthine can't get in. So, their conversion to uric acid slows down or stops. This is called product inhibition. Now let's talk about indirect inhibition. You see, inside the liver or any other cells, both synthesis and degradation of enzymes happen continuously. Old enzymes get degraded and new enzymes are synthesized. Now, there are some drugs like ethionine, puromycin, acetinomycin D. If a person takes any of these drugs, they inhibit the synthesis of new enzymes required for the degradation. With less or no new enzymes, less metabolization of drug happens and drug molecule level rises inside our body, ultimately causes serious effects. There are some other drugs like carbon tetrachloride, carbon disulfide and disulfiram, etc. What they do? They increase the degradation process of enzymes. Above drugs inhibit the synthesis of new enzymes. These drugs promote the degradation of enzymes. So enzymes get degraded quickly. So metabolizing capacity decreases. This decrease in enzyme content, either by promoting degradation or inhibiting the new enzyme synthesis, is what we call repression. Indirect inhibition happens by one of these two mechanisms, repression, and the second one is altered physiology. Now, let's see about altered physiology. Due to nutritional deficiency or hormonal imbalance, enzyme inhibition happens. This is an indirect inhibition mechanism. That concludes our video. Our next topic is inhibition of biliary excretion. We will continue this topic in our next video. Thank you for watching. Yours Pharm.D Guru signing off.